Over the past 10, 15 years, we've just seen an exponential change in our ability to monitor and perturb neural circuits using viral vectors to insert proteins from other animals or synthetically generated proteins that allow us to control neural activity with millisecond precision. And using these tools, we can actually control neurons in a certain part of the brain. We can control neurons that project from one area to another. We can control neurons that express a specific biomarker, like a receptor for dopamine or serotonin. And we can even control neurons based on their pattern of activity. Researchers at MIT have actually used this tool to turn a positive memory into a negative memory in mice. And researchers at Princeton have used a similar technique to take a mouse and teleport him. Basically, observe the pattern of activity when a mouse is in one place, and then when he's in another place, they replay that pattern, and the mouse thinks he's in the original place. So it's really like science fiction right now. And these tools have allowed us in my lab to use cheap electronics to build a high-throughput training center where we're currently training about 100 animals per day. And one person, one technician, can train 100 animals each day. And this is important because if we want to study complex cognitive tasks, we need a lot of animals. It also helps us study individual differences. And this is another thing that's really been missing from neuroscience. So these new tools really allow us to link DNA to proteins to neural circuits into behavior. And these genetic engineering tools probably won't be used in humans for many, many years. They're just not that safe yet for human use. But what they will allow us to do is identify specific neurons involved in some behavior or some disease. And once we know which neurons are the key neurons involved, we can then use pharmacological approaches to find safe ways to manipulate those neurons, which could be directly used in humans.